Hi guys, we're here to talk about evidence for evolution. And so one of the evidences for evolution is direct observation. We actually see it happening. Now again, if you're thinking of evolution as a speciation event, humans breaking off from a common ancestor from monkeys, we didn't see that happen. We're not going to see that happen because it happened before we were humans or as we became humans. What we're seeing are the microevolution. That's what we can observe. That's what we can have data and evidence to show is happening, okay? And so when I'm talking about these different things, what we are seeing is the accumulation of microevolution that led to macroevolution, but we don't necessarily see that macro. Sometimes we'll see after it's happened, we'll see, oh, this was one species and now it's two. But for the most part, we're seeing microevolution. So the first is I can give an example of direct observation as support for evolution. One of the examples is Endler's guppies. And so Endler was a scientist and he studied guppies. And he noticed that in different areas of streams, which is where guppies live, the guppies had different colors. In some areas they had bright blue spots, and in other areas they had more drab coloration. And so he did a lot of research, took pictures and collected data, looked at the predators, looked at the color of the rocks on the bottom of the stream, the gravel at the bottom of the stream, looked at population numbers of the guppies, those kinds of things. And what he realized, or what he came to the conclusion, was that guppies have to strike a balance. Guppies have to survive, right? The whole goal of living is surviving. And so they can't be too flashy or they're going to attract predators. Predators are going to see them against the gravel backdrop of their stream. However, they do have to attract ladyfish, lady guppies. And so they do have to be flashy enough to stand out from the other males. And so what he would see is in areas where there were lots of predators, the guppies tended to be more drab. There tended to be less coloration because the survival risk was too high. So yes, they did want to reproduce, and yes, flashy colors make that more likely, but there were too many predators, the survival risk was too high. In areas where the gravel was very varied, like there was a lot of different color gravel, and in areas where the predator populations were low, he saw more flashy guppies like this guy. So lots of different colors because the gravel is different colored, he's more likely to blend in, and also because there's fewer predators, the survival risk isn't as high, so they don't have to worry about getting eaten as much. So they can afford to be a little bit flashier. Yeah, some of them are still going to get eaten, but others will not. So that was one example. The peppered moths are another. So I like this picture because it shows you the tree with soot on it versus the original tree. So this is a birch tree, and you can see here's another birch and how the peppered moth blends very nicely in with the birch trees. And so this is pre-industrial revolution, or maybe now that it's cleaned up again. And you can see the lighter colored moths blended better. During the Industrial Revolution, where all the soot was on the trees, you would be able to see those light-colored moths really easily, and the dark-colored moths would have blended better. And so what we saw is during the Industrial Revolution, the numbers of light-colored moths, which were higher, fell, and the numbers of dark rose. Okay. Then after the Industrial Revolution was over, but when they started putting scrubbers and things like that on industries to clean the air before they release it, and the soot levels dropped and the trees went back to their normal color, they started to see that shift back to more lights and fewer darks. And it's all about what the birds that eat these moths can see. If I'm flying way up above a tree, this is the one I'm going to see on that dark tree. I'm not going to see the dark ones. They're going to blend in. And so when I'm a bird looking for food, I'm going to go for the ones that I can see more easily. Okay, so those are two examples of direct observation. Antibiotic resistance with bacteria is another one. Insect resistance with DDT is another one. So those are some other the things that we have actually been able to see these evolve. All right, the next piece of evidence is fossils. I can explain how the fossil record supports the theory of evolution. So the fossil record does a couple things. It allows us to see organisms that are no longer here, which allows us to see a lot of things that we otherwise would not have seen. It also allows us to see the progression. So this is the progression of horses. This is a horse today, Equus, and you can see they're really zoning in on their legs, on their feet. You can see they were multiple-toed, and then over time those toes fused into their hoof. They have multiple hooves. They only have one hoof. Anyway, so you can see the horse has also gotten a lot larger over time. And how do we know this? We know this from the fossil record. So we've seen this evolution, this change in the species over time by looking at the fossil record. It also allows us to see, how did we get from fish to amphibians and reptiles? Well, the fossil record has kind of given us some clues there. All right, the next one is I can discuss the idea of biogeography as evidence for evolution. So biogeography is the study of where things live, basically. And so when they studied, they found these fossil remains of Cynonathus, I don't know how to say that, this dinosaur little lizardy thing. And they found them in the middle of South America, as well as kind of the southeastern part of Africa, maybe not quite southern, but middle eastern part of Africa. And they didn't really understand why. Why did we see the same fossil bones here as we do here? Well, once they got into the idea of continental drift and figured out the whole Pangea and all of that kind of stuff, then they realized, oh, this is why, because this used to be connected and this little guy just walked back and forth. I know we show it gapped here, but that's just due to erosion and things like that. They were literally connected. We also see this brown swath of this Lystrosaurus. Okay, India was actually nestled up against all these. It floated straight up through the Indian Ocean and slammed into Asia and created the Himalayas. Anyway, we see this fern. We see this fern fossil all over. And so once you know about Pangea, these patterns make sense. They did not make sense before. Like, why on earth is this found here in Australia, here in Antarctica, here in Africa, India, South America? What? But now we realize, oh, because those all used to be together. And so it does support, I should say the fossils support plate tectonics. But as far as evolution, another thing we can look at is we can look at species. And species A blew over from the continent over here, and so it's very similar to the continent. But over time, as the island broke up, the species evolved into A, B, C, D, and E. Okay, so mutations, these populations got separated. Mutations occurred that allowed each population to become slightly different than the other. However, all of these, let's say we're talking about Galapagos finches. So let's say this is the Galapagos Islands, and these are all finch species. If we look at the species of finches over on South America, these species are all very similar versus the finches you might see in North America or other parts of the world. So biogeography allows us to look for relatedness due to where species live. Okay, the next is I can discuss morphological or an anatomical homology and how that is evidence for evolution. And so when we talk about homology, we're talking specifically about homologous structures. So this ties back to divergent evolution. And so why do we all, like if I compare my arm to a bat arm, we have a humerus, we have a radius and an ulna, we have carpals, metacarpals, and phalanges. And the reason we have these is because of the Hox genes. Sorry, I have a yawn there, I'm trying to suppress. The Hox genes for these are in our genes. So it's our body tells us, the Hox genes, if you remember those homeotic genes, are the master control genes on how you set up your body. And so large structures like your bones and things like that are coded for in your genes. So why do I have a humerus and why does a bat have a humerus? Because we have the gene that says make a humerus or make a radius or whatever. And so that's why we have it. Well, where did we get that? We got those genes from our ancestors. And so when you see bones and structures that are similar, it either means convergent or divergent evolution. And so once you rule out convergent and you realize that they are not distantly related, they're actually closely related, then you know it is a homologous structure. Okay, another type of homology we can look at is embryology. So embryonic development is also coded for by your genes. Your genes tell your body when to turn, your master control genes like Hox tell your body when to turn certain genes on and off. And so your eyeball genes cut on at a certain time and that made your eyeballs. And then they cut off because you didn't need any more eyeballs and things like that. So what we notice is the genes for very early development are very similar for fish, salamander, tortoise, chicken, human, right? We all look, there's certain things we have in common. We have big old knobby heads, we have tails, we have these little gill slits, these little pouches that for some become gills, for others they disappear. But there are for sure some similarities. We're curved kind of in that fetal position, you could say. Okay, so that tells us the genes that told for all of these things, these eyeballs, the tail, we all have those genes, okay? What's different is how those genes are controlled when we want to turn those genes on and off. And so what we see is as you get further and further into development, they become more different. The fish and the salamander stay the same almost to the end because a salamander is an amphibian, and so it is very similar to a fish. If you look at, say, a reptile, then you are looking at the similarities between a chicken because birds are actually reptiles. Sorry, I'm checking out one thing real quick. Birds actually evolved from reptiles, I should say. It's probably a better way of saying it. So that's why you see a lot of similarities between a turtle and a bird, because they are, even in the stage 3, look at, I mean, they still have that kind of beak-like structure, because turtles have, they do call them beaks, but they're not like a bird beak. They have little fat bellies, right? Of course, turtles have a shell. They keep their tails. So there's a lot of similarities. Of course, birds start to form wings. And then, of course, when you look at the human, we start to change a little bit in this mid-stage because we're different. But if you compared us to another mammal, like a monkey, you would see that we'd be very similar, or a rabbit or a pig. You would see it very similar in this mid-stage. And so the longer they stay the same, the more genes they have in common. So we would say that the fish and the salamander have the most genes in common because they stayed the same the longest. The turtle and the chicken have a lot of genes in common because they stayed the same the longest. But we obviously don't have a lot of genes in common with a fish because by this mid-stage, we're very different. And so that tells us that the genes are different. Okay. The last one is genetic or molecular homology. There we go. And so what we see, oh, I didn't mean to close that one. What we see with that is we see a comparison of either the DNA or amino acid sequences. And so when you look at the DNA or amino acid sequences, this is amino acid, what you're looking at is areas that they're conserved. So it's the same. Those are because those are probably so important, they would change the function to the point that it would no longer work. And areas that are not conserved. So in cows, rats, and mice, there's a P there, which is probably phenylalanine, and then humans and chimps have a lysine there. And so this is coding for the protein that your DNA wraps around. So some parts of that protein are super important, and so they are conserved across the board. Other parts are not as conservative, or not as important, and so they're not conserved because they won't impact the function of the protein. And so when you see those differences, you see that the human and the chimp have basically no differences because our DNA is so similar. Another way you may see this presented is like this, where you have the number of DNA differences, the number of similarities, and the percentage difference or something like that. You might just see percentages, you might just see the differences. The more differences, the less similar they are, and the more long ago, that's not good English, the longer ago they broke apart. So the fact that we are very similar to a chimpanzee tells us that we broke apart fairly recently from the chimpanzee, whereas we broke apart a lot longer ago from an orangutan. Now, if you had something like a fish or mold, so fungus, you would see even more differences because we broke away from those even longer ago. And that is molecular homology. So I think that's all of the evidences and how they support evolution. The evolutionary theory, I should say. But if you still have questions, please let me know. Thanks, guys.